Great. So while Chris is getting that loaded up, we'll do a quick introduction. My name is Alex Falsey, and I am the Clinical Research Coordinator for a genetic study at the Children's Hospital of Philadelphia under the leadership of Dr. Nancy Spinner.
And my name is Chris Krachowski, and I am the Senior Research Technician in Dr. Spinner's lab.
So as Dr. Piccoli introduced yesterday, Dr. Spinner has been working on Alagille syndrome for a number of years now, and actually her lab was the one to find both JAGED1 and Notch2 and their role in the Alagille syndrome. But one of the questions that we have not yet answered, and we're still trying to figure out, is how is it possible that individuals within families can have the same JAGED1 mutation but can have very different cases of Alagille syndrome? So in genetics, we call this variable expressivity. So the question is this just a random variation? Is this just something that happens? Or are there other genes that could potentially be involved in the pathway and in terms of the clinical outcome? So to try and answer that question, we have looked at our wide repository of samples from people with Alagille syndrome, and we're curious to compare individuals with JAGED1 mutations and look across the rest of their genetic code, the rest of their genome, to see if there are any other regions that could contribute to their liver disease severity.
Okay, so to do this, we did something called a Genomewide association study, and that basically means that we took two groups of people. So in this case, we took individuals with mild liver disease and individuals with severe liver disease, and across both groups, they all had JAG1 mutations. And so what a Genomewide association study does is we want to see what's the difference between the two groups. So we know they all share a JAG1 mutation, but are there other regions of the genome that they have in common or different that the two groups share together? So when we did that, we ended up finding a region of the genome that was close to a gene called thrombospondin 2, and this gene has known interactions with the Notch signaling pathway. So then we wanted to see what is thrombospondin? So it's a gene that is affected in cell adhesion and migration, and why we originally wanted to zero in on this region is because we know that it has known interactions with the Notch signaling pathway. And so we kind of wanted to look more into how this gene might interact with the JAG1 mutation or interact to create either a mild or severe liver phenotype. Okay, so our first question that we asked is thrombospondin in the right place? So if it's expressed somewhere outside of the liver, that's not as interesting. So we wanted to see if it is expressed in the liver, and specifically if it's expressed in portal tracts or mouse bile ducts. So we started in the mouse, and we wanted to do a staining. So I know it's a little bit hard to see. But basically, we were able to see that thrombospondin is present in mouse bile ducts. So from there, we wanted to move on and see more specifically if it's a different expression in the mouse portal tracts versus the rest of the liver. So we did a really cool experiment where we took a mouse cell, so a mouse liver cell, and we laser captured out just that portal tract. Okay, so we had just the portal tract, and then we had the rest of the liver cell, and we wanted to see, is there an increased expression in thrombospondin in that portal tract versus the rest of the liver? So when we did that, we basically saw that there is about an 8-fold increase in expression in the mouse portal tract versus just the rest of the liver cell. So we know that thrombospondin as a gene is in the right place in high levels.
So just to take a step back and provide a little extra context, when you do have all of your genes, you have about 20,000 genes in your genome, so all of your genetic code. And not every gene is expressed in every place in the body. So as Chris was saying, it's particularly interesting that this thrombospondin 2 gene is expressed around the portal tracts in the liver. It gives us an indication that this could play an important role. So we've done a lot of studies in the mouse samples, and so this is where you step in and hopefully can help us out. So we really are interested in seeing how does thrombospondin 2 express in human samples. So unfortunately, thrombospondin 2 is not expressed in blood. It just isn't. Unfortunately, the only place it is expressed is in fibroblasts. So that would require us to obtain skin samples. And actually, Dr. Lin is here, and he can help us. So we've got a sign-up sheet outside. It's really not a painful process, and this could help us to figure out what role this plays in human disease. So if you have any questions, we are available. I promise we don't bite, and we're pretty good at answering any questions you might have. So please don't hesitate to ask us.
So just going back on that, So I tried really, really hard to get this to work in the blood. It's just not expressed in the blood, and so I can study a mouse sample as much as I want, but it's much, much, much more interesting to look in humans. And so getting that skin fibroblast sample is really, really important because it allows us to see specifically the people who have JAG1 mutations, how is thrombospondin look? How is it expressed differently in different people? So it's really, really, really important that we can get fibroblasts specifically. So fibroblasts are just skin samples. So it's really, really easy, and it would really help the research a lot. So just on a completely different path, so one other pilot study that we've just launched in the lab is to do whole exome sequencing of people who are JAG1 and Notch2 mutation negative. So whole exome sequencing is a relatively new genetic procedure that allows us to look across someone's entire genome at every single gene. So if you think about JAG1 and Notch2, so those are two genes that everyone has, but people also have about 20,000 other genes, a little more. And so whole exome sequencing allows us to look at every single one of those genes. And so since 4% of individuals who are clinically diagnosed don't have JAG1 or Notch2 mutations, doing whole exome sequencing allows us to see every other gene to see if they possibly have mutations in anything else. So again, we've launched a pilot study. Right now, we've done about 3 families, and we are in the initial stages of validation to look at some very interesting variants that we found.
So I think that sums it up. I know this has been very science heavy, but please don't hesitate to come ask us any questions. We'd love to break it down for you and explain a little bit further. But we are here on behalf of Nancy Spinner, who unfortunately couldn't be here this weekend, but she was very sad to miss it. So our team really is very heavily populated with some amazing people. So Nancy Spinner, Kathy Looms, who also couldn't be here, David, who's sitting back there, Marcella Devoto, and we definitely couldn't do this without support from the NIDDK. So thank you so much, and please don't hesitate to reach out.
So we've had a request here really quickly to have Chris just tell you what a skin sample involves.
And again, I'm totally willing to, if anyone wants to see, I'll do one on my own, or Henry will do one on my own arm. It doesn't hurt at all. It's completely numbed, and it's just a tiny little, it's less than a fraction of your fingernail. It's just a small little punch right here. It just takes a tiny skin sample. So more detailed information, I think Dr. Lin would be able to explain.
Good morning. Pleasure to meet everyone. Sorry I couldn't make it last night. The skin biopsy stuff sounds pretty intimidating and ominous, and I agree. I actually thought the same thing the first time I did one. But we've been doing this for quite some time now, and actually what it entails is, as Chris was describing, it's a small punch biopsy. Essentially, it's less than half a centimeter across in diameter is about how big of a biopsy or hole that we're making in the skin. It's not very deep at all. It's probably about the same amount deep. So if you think about it, it's like 1/4 of an inch by 1/4 of an inch tops. It's a small little circle that actually, you know, what happens is we numb up your arm. We take it kind of in the fatty part of the arm right there, or right here. I don't have any fat. Hey.
That's okay. We're friends.
So you take it in the fatty part of the arm here, you numb it up, and then you take the skin biopsy. It's actually such a small biopsy that you don't even need a stitch to sew it up, and actually you put a Band-Aid over it with Neosporin, and it should heal up. But I think that we have a few comments, questions? Yes, at the picnic. Yeah. There you go. Oh, so we just had a few of our prior skin biopsy volunteers who are volunteering to show the scars if there's any questions, actually. Yes. Yes. Needle for the numbing.
And again, the more samples that we get, the more interesting conclusions that we can draw from our study. So really, really like as many people who are willing to participate to hopefully